So we're going to look at a little bit more detail about mitosis, and first we want to know kind of where we're going. So the basic idea is that we've started in G1 with individual chromosomes. Those individual chromosomes are going to be duplicated or replicated in S phase. And those two copies stay attached to each other at the centromere until partway through mitosis. And the mitosis is going to take those two copies, divide them as well as the contents of the cell, and make two individual daughter cells that are genetically identical. So this is an overview of the beginning of mitosis, and it starts really with G2 of interphase. We know that by that point the cell is committed to cell division. And these are actual pictures. They're not really this color. Those are fluorescent dyes. But these are real cells. And what you can see in the bright yellow area is the formation of the centrosomes. And these are the structures that are going to organize the cell skeleton materials that will pull the chromosomes and pull the cell apart. So we can see a cartoon of that, the centrosomes, each with a pair of centrioles. The chromosomes have been duplicated, but they're still kind of an unwound mess in the nucleus. The nucleolus is still visible, and the nuclear membrane is still there. So prophase is the real beginning of mitosis. And this is when the chromosomes condense, meaning that they are packed together. They need to be untangled and packed tightly to prevent breakage during cell division. And this is the point in the cell cycle when you can actually really see the chromosomes for the first time. You could tell where they were before, but you couldn't see individual molecules, and now you can. And that's why, as I noted, so many microscope images of chromosomes have both of those chromatids visible. At this point, the nucleoli will disappear, and the centrosomes will start to move to the ends or poles of the cell. And you can see the two little yellow centrosomes here. They're moving around. And microtubules will start to attach to those centrosomes, and these will become the sort of structural scaffolding for the cell and for the chromosome movement. So these microtubules are also referred to as spindle fibers. The next phase is prometaphase, which, as you can imagine, is between prophase and metaphase. And here we're really going to see those chromosomes very clearly as individual molecules. The centrosomes are completely at either end of the cell, basically. And the microtubules will start to attach to the chromosomes at their centromeres. And that structure is called the kinetochore. And the kinetochore is a protein complex that forms at the centromere of each chromosome. So you've got two types of microtubules in here. Microtubules that are attached to the chromosomes via the kinetochores, and then microtubules that are really just pushing against each other, providing structure for the rest of the cell. The next phase is metaphase, and this is one of the most recognizable phases of mitosis. The chromosomes have been pulled by the microtubules to line up across the center of the cell. And so you can see the chromosomes kind of across the middle here. They look much more complicated than they do in this nice diagram, and that's because chromosomes are long, and each of these tails can sort of flop around individually. Remember that they're only held by the microtubules right at the centromere. So it doesn't look quite as pretty as the cartoons draw, but you can see that those chromosomes are lined up across the midline or the metaphase plate of the cell. Interestingly, it's been demonstrated that each chromosome needs to be attached on both sides in order for this system to work and in order for the cell to continue to move forward. So each centromere is pulled on both sides by microtubules from either end. The next phase is anaphase, and you can see that at this point, the proteins that are holding the centromeres together have detached from each other, and one sister chromatid is going one way and the other sister chromatid is going the other way. So if we look at this in cartoon form, it's a little bit easier to see. each of those pairs of sister chromatids that were held together at their centromere are being pulled one to one side and one to the other side. And now we would refer to these as individual chromosomes because a chromosome is really defined by the presence of that centromere. So at this point, the cell starts to elongate. So these microtubules, non-kinetochore microtubules, are giving structure to the cell and making sure that those two poles don't collapse together. And each cell will then get one copy of each chromosome, forming two genetically identical cells. The last part of mitosis is telophase, and this is when the daughter nuclei form and the nuclei will reappear. Nucleoli will reappear. Cytokinesis is actually the process of physically dividing the cell, and sometimes people think of that as part of mitosis. Sometimes you'll see people refer to mitosis and cytokinesis as though they were separate processes. Technically, mitosis is the division of the genetic material, and cytokinesis is the division of the cellular materials themselves. So the basic idea is the nuclear membrane starts to reform on either side. You've got one centriole on either side, centrosome, and the cell sort of pinches off in the middle, again driven by microtubules. So this is a bit about the way that those microtubules behave. This actually shows a pretty cool electron micrograph of the microtubules attached to the kinetochore. So you can see that the proteins really pinch in those chromosomes and the microtubules attach to those proteins. There's been some interesting work done looking at how microtubules actually pull those chromosomes around. So one experiment was done was to mark a portion of the microtubules and try to see where this dye line moved as the chromosomes moved. And what they found was that the dye line stayed an equal distance from the centrosome, but the distance between the dye and the chromosome got shorter. And what that indicates is that the microtubules are polymerizing and depolymerizing at this end, the end that's attached to the centromere of the chromosomes. And motor proteins, it appears, are pulling the chromosomes along those microtubules. And the tubulin is polymerizing and depolymerizing at that end. So cleavage in animal cells is a little bit different from cleavage in plant cells. We don't even call it cleavage in plant cells because the structure of the cell is inherently different. Animal cells are flexible. Microtubules and microfilaments pinch off the middle of that cell to form those two daughter cells. In a plant cell, you've got the cell wall that's giving the plant cell shape. And so what happens is you get this row of vesicles that are formed along where the two cells need to divide. Those vesicles actually secrete the materials that form the cell plate, which becomes the new cell wall. Cell division in bacteria is not surprisingly a little bit simpler. It's referred to as binary fission. Bacteria have a single circular chromosome, and so basically what happens is the chromosome is copied. The two origins of replication, where replication of those chromosomes are started, end up on opposite sides of the cell, and then the cell sort of splits in the middle. It's a fairly simple process. There's some interesting hints about how mitosis evolved, looking at dinoflagellates and diatoms as well as yeast is what's shown on the bottom here. Different structures of microtubules, different processes for actually splitting the chromosomes, suggesting obviously that there were multiple steps in the evolution of this complicated process, not surprising. So that concludes our video on the cell cycle and mitosis. In the next video, we're going to talk about the control of the cell cycle.